Assalamualaikum and good day everyone. My name is Nur Amnani Shara binti Lawrence. Today I would like to present my individual project for AGA402 in Principle of Nursery Management entitled Capsicum Fruit Trescents for Propagation. In this project, I carry out the propagation process of capsicum fruit trescents, also known as ladabara, and observe its growth over the period of 5 weeks. This is the plant that I selected for my project, which is Capsicum frutescens, or commonly known as Ladabara. I chose this plant because it is highly suitable for cultivation using a fertigation system. It also has a relatively short growth cycle, making it suitable for observation within 1 semester. Another reason is that this project allowed me to apply nursery management practices that I learned in class, such as seed propagation, seedling care, irrigation, fertilization, and pest management. In addition, my family operates a commercial fertigation farm, so I had the opportunity to gain practical experience in the real farming environment. These are tools and materials used throughout the project. First is the seedling tray, which is used for sowing seeds. Next is the certified chili seed, which ensures good germination and healthy seedlings. I use peat moss as the growing medium because it retains moisture well and supports seed germination. A watering can was used to water the seedlings regularly. The AB solution was supplied as fertilizer to provide essential nutrients for plant growth. Finally, a plant sprayer was prepared for applying pesticides if pests or diseases appeared. The total cost of my project was zero. This is because all of the materials, equipment, and facilities were already available at my family's commercial fertigation farm. Therefore, no additional purchases were required during the project. During the first week, I started the propagation process using the sexual propagation method, which involves seeds. First, I prepared the seedling tray and filled each cell with peat moss. Then I sowed one seed per cell before watering the tray carefully. After that, the tray was placed inside the nursery under suitable conditions to allow the seeds to germinate. 5 days after sowing, most of the seeds had successfully germinated. The seedlings emerged uniformly and looked healthy. There were no visible signs of pests or diseases. The successful germination showed that the growing conditions, including moisture and temperature, were suitable for the seed development. In the third week, the seedlings continued to grow well. The first true leaves had developed and the stems become taller and stronger. Regular watering and proper nursery care help maintain healthy growth. No pest or disease infestation was observed during this stage. By the fourth week, the seedling had become larger and stronger. The root system was well developed and the seedlings were almost ready for transplanting. The plants continued to grow uniformly, showing that the propagation process had been successful. In the fifth week, the seedlings were transplanted into polybags. The transplanting process was carried out carefully to avoid root damage. After transplanting, the seedling remained upright and adapt well to the new growing medium. This indicates that the seedlings were healthy and ready for the next stage of cultivation. In conclusion, this project successfully demonstrates the sexual propagation of capsicum frutescens Throughout the 5 weeks, I observed the complete process from seed sowing, germination, seedling development until transplanting. This project has improved my practical knowledge and skills in nursery management, especially in plant propagation and seedling care. Thank you for your attention.